Hello, everybody. Welcome again. And I would like to say that if you are watching my videos and you are not subscribed yet, oh my god, shame on you, right? Please subscribe and come with me because from now on I will start recording a DOE series, okay? The first one is about how to perform a very practical analysis when we have data from our DOE, something that sometimes was very difficult to collect, very difficult to obtain. But now we have the data and we have to start analyzing this DOE, our design of experiments, and there are some very simple tools in JMP that we can use to understand how good or how bad was our results. Okay, I have here two different data tables. This one is about a fractional factorial DOE with 8 different factors, one response variable. There's no replicates here. There's nothing different, unusual. All right. This is one of the most famous DOEs in the world. 8 factors, 16 different combinations, the 16 runs. And the left side here, I have a DOE with 4 factors, 16 different treatments that we say here. It's a full factorial DOE with all the possible combinations that we call treatments. And for each treatment here, we have 5 different parts. Okay, so we replicated some parts for each combination. Okay, so I will analyze. I just did something here because I was recording another video in Portuguese before and I forgot to clean, to reorganize the data table to start again. So now you have a spoiler that what I'm going to do here, right? So I will start here with this data table, and one of the first thing that we can do here is organize this response variable here, sorting by ascending. So now we have all the data here sorted, and we will use a graph here inside of this legacy called overlay plot. We will put the Y here and click on OK, and now we have all our data from our DOE organized from the first one to the last one in an ascending order. We can connect adding line here, and now we can see, we can observe that if we had some special calls, if we have a variation that is it's an over variation here, that maybe we should transform this data or not. So this is a very simple graph that we can put our specification limits here and understand if we are good or if we are bad. Okay, this is the first one. The second one is our variability. So putting the Y here and the pattern here, we can understand how was the result of our DOE by each different combination of the signals of our factors. So the combination plus 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 plus, we have this value, and the minus 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 minus, we have this value. We have some values here. We can analyze it and see in other perspective how good or bad were our results. Okay, so this is the practical, very practical analysis in our DOE. Okay, don't save. Let's talk about this data table where we have 80 rows because we have 5 different parts measured by treatment, by combination of factors, and we have a full factorial with 4 different factors. The first thing that we can also do is organize this, sorting by ascending, performing exactly the same graph, overlay plot, putting the Y here. Now we will have 80 points. We can add a line and we can see how was our variation, how good or bad. If you put the specification limits here, you can have better reports. So this is the first one. The second one for me is more important, is a variability. Mainly when we have a DOE that was replicated, okay? Why? Because here, guys, we can see the variance between the different treatments. The variance if it was constant or not constant. We can use this graph here. This graph is talking about the standard deviation inside of each one of these bars here. So we can see that some treatments we have more variation than other ones. For example, this one we have few variation. It can mean something important for us. Maybe this is your goal or not, so you can put the specification limits here and we can understand how good or how bad was the variation between treatments and variation within treatments. Okay, you can put specification limits, you can change the colors here. For example, let's suppose that we cannot have parts above 11.05, so everything that is above this value we can just change the color of the rows, and now we can have better reports in our presentation. Okay, so this is a very practical analysis for DOE. I hope you enjoyed this video and be prepared, wait for the next one when we will talk about some graphical analysis. Okay guys, hope you enjoyed, subscribed, and see you in the next one. Bye bye.